So today we are going to look at ImageJ software to examine how we can use the software to identify different morphologies of cells using the capabilities that are present within this system. So we are going to do actually two things. First, we are going to learn how to identify the different morphologies, and then we are going to go back and we are going to count the number of cells that are in each one of those morphologies. So there are two different ways to do that, and I'm going to show you kind of the rough and easy way first, and then I'm going to show you the more detailed, more quantitative way. So as you know before, we have used ImageJ to identify, to keep count of different types of cells in an image. However, usually in that case, we were only keeping count of one morphology at a time on an image, and we were not keeping a track of all those different morphologies at the same time. So today we are going to use a way that we can keep track of all the different morphologies in the same image. So here I have an image opened up in ImageJ. This image is from a mouse meioblast cell culture that was in differentiation medium for a few hours or for a day. So they are starting to differentiate into myotubes. Now they are way far away from being fully developed myotubes at this time, but you can see that some cells are still looking very similar to how they look when they're fully undifferentiated, which is, kind of circular or boxy. And then other cells, so this is still, looking very kind of boxy cell, while other cells are becoming more elongated and are beginning to look a little bit different in their morphology. So here you can see, for example, this cell is nice and long, it's getting thinner, and it's definitely showing us signs of different morphology in them. So if you look around, you'll see many cells in the central cluster that are exactly exhibiting that type of morphology, where they're beginning to differentiate and are getting ready to merge with other neighboring cells to form the myotubes, while other cells are still the same boxy, undifferentiated ones. And then if you look closer, you will see that there are other cells that are kind of on the borderline, right? So if you look at this one, for example, this was not a very good drawing from me, but this one is still kind of boxy, but at the same time it does seem to be getting a little bit pointy on the edges. It's beginning to get that elongation, but we don't know where that would fall. So we maybe wouldn't know exactly which category to put it in if we just had cells that were undifferentiated completely and cells that were beginning to be elongated and showing signs of differentiation. So the first thing that we will do is just basically look at them by eye and categorize them into those two categories: cells that are boxy and cells that are and fully undifferentiated, versus cells that are elongating and beginning to show signs of differentiation. So we are going to use this multi-point tool like before. You can change it to show itself differently, so you can make it into a cross or a dot or a circle. So if I click cancel here, and if I click on one of these, I can change that to a hybrid or just a dot or just a circle, whichever way I want. So I'm actually going to go ahead and make that into a cross at this time for this first run. And now you can see that it says label points, so you can see that, and the counter says 0. So keep those two things in mind. And I'm just going to click on OK, and I'm going to go ahead and click on all the cells that I believe are undifferentiated at this time. So any cell that I am certain can be counted as an undifferentiated cell, I'm going to go ahead and I'm going to mark them with my little pointer. So some of the cells, if I'm unsure about them, I will not use, but otherwise I will go ahead and click on all the cells that I've thought were undifferentiated in this image. Okay, so once I have done that, I am now going to go ahead and go back to this tool, and I'm going to go to the counter, and I will go to the next number and click on 1 and click OK. And this time I'm going to go ahead and start looking at the numbers that are, or looking at the cells that are definitely elongating and definitely showing signs of differentiation. And I'm going to go ahead and start clicking on those at this time. So as you can notice that now it automatically changed the color of the first crosshairs, and it gave them all a 0 instead of the number label that they had. So before it was counting them and showing the counts. Now it's not showing the counts, and instead it's just showing a 0, letting us know that they were all categorized similarly by the system, or by us in this case. And now I'm going to go ahead and do the same thing for this one. I will mark any cell that I know for a fact is showing signs of differentiation and can be marked a differentiating cell. And so, let's say that these were all the cells that I was sure about and that I fully believed were differentiated. And so you still see there's some cells that are not marked either in either category. So those are the cells that we now have to worry about. So we can go back again into this crosshair, and again you notice there's a number at the bottom. That's the number of cells in that category. If we were to go back and see 0, it will show that number as well, and it will say, okay, so now you can write that count down, for example, and say there were 21 undifferentiated cells. There were 26 that were showing signs of differentiation. And now I'm just going to go to 2 and mark the cells that I am unsure about or that were hard to detect. And again, it's going to do the same thing. It's going to let me place a third color on there to depict these cells that I am unsure about, or that I cannot count appropriately given the circumstances. Maybe the lighting isn't right, maybe I can't see the cell completely, whatever the reason may be. I can now label them so I know which cells were omitted from the counts for whatever reason that I had. Now some of these cells may actually be able to, we may be able to categorize using our alternative approach, which I will show you in just a second. So now I have marked these, and I can go in and I can look at that count, and it shows that there are 13 cells that I have not been able to categorize through this count. So that's how you would go ahead and you would make your counts. And now you have a single image that shows all your different morphologies and how you categorize them on the same image, in the same place. So for your own record, whether it is for your research needs or for your, maybe you're taking a course where you're doing this, maybe it is to show your instructor how it is that you're categorizing these cells, this is what you would get. Oh, there's another one right there that we didn't count as well. So what you would do at this point is go ahead and write down these numbers for yourself in your whatever data sheet you have or an Excel file that you have for each one of the categories. You would say that there are 21 in category 0, 1, 26 in 1, and 14 in 2. And then you would take this image and export it out with all this drawing. You could also just simply do a print screen of this. And then, so I could just do a print screen, and I could put that into a PowerPoint slide, for example. So I have a record of that for myself as well. Sometimes it's easier to do that simply because the images that are taken through print screen are less bulky. Imager usually gives a really bulky file otherwise, and so it can cause storage issues for some people on their devices, but having a print screen image is usually less bulky. It's not as high resolution, but it provides all the information that you need. And so, especially for my students in research, I ask them to make a slide for each one of their images when they're doing this, and then insert a text box with all the counts on the side so that they have that information handy and available right there for them. So I'm just going to do that actually right here, text box, and I will say this is my control set. It will show you the image number up there anyway, but obviously you want to make sure it's my control C3B image, and it had 21, and then so I could write that right there. And you could also place in the beginning of your file, some information about which one is which, a legend, so we know which numbers are which. So this is your undifferentiated, your pink one is the differentiated, and blue 2 are unclear or undetermined. Okay, so now you have record of all your cells and the way you had them. Now we get to the second part of this exercise, where we want to now also determine these cells that we weren't sure about. How would we do that? To look at some of these unclear cells, we need to first think about what type of differences are present between a cell that is in one category versus the other. This one is a very clear difference. The cells that are undifferentiated are more round or boxy, versus the cells that are differentiating, those are more elongated, so they would be less round. So if we were to go under Analyze and go to Set Measurements, you would find that there are lots of different parameters that we can choose from. So I could say in here that I wanted to look at skewness of a particular sample or a particular cell. I could look at whether it fits an elliptical form. If they are, there's usually a thing called roundness, if they are round or not. And those can help me figure out if the cell fits a one category versus the other. So I'm going to do a shape descriptors and a fit ellipse for my choices, and I'm going to say OK. And I'm going to make it into 1 decimal places just to keep it simple. So let's see what that does for me. So now I'm going to use my PowerPoint image as a guide. Actually, I'm going to go for a second to this one. And I'm going to go to the selection, freehand selection, and I'm going to go ahead and draw my boundary around one of these cells. So this was one of the ones that we know was fully undifferentiated. And I'm going to go to Analyze and Measure, and I now see that it gives me a round, an AR score, a solidity, circular, how circular it is. So it gives me some information about this cell. I'm going to now repeat that for a cell that I know for sure is differentiated or is differentiating, like this elongated cell or this elongated cell. So let's go ahead and do this one, for example, which is nice and long. So this will give us an extreme points for our stuff. Again, drawing the outline, drawing along the perimeter, and click measure. And now you can see how the roundness for a cell that was differentiated was 0.8, and for one that was or undifferentiated, and for one that was differentiated, that was elongating already, was a lot lower. You can see the same kind of ratios in your circularity as well. So we could create some arbitrary cutoff numbers to decide which are these cells that we are unclear about. So one of the things that we can do is we can go ahead and measure every single one of these cells the same way. And this would be a more quantitative approach to our problem. We could go ahead and we could do this for every single cell that's in here, right? And we would just keep going and say measure. And, normally you would want to be obviously go in an organized manner so you can keep track of these cells and not double count them. You could also mark them as you go, as we've talked about before, to keep track of them so you don't double measure the same cell. And if you have forgotten how to do that, I can show you in a second. Measure right there. For example, if we wanted to keep track of it, you could go to noise and add noise, and it kind of fills it in. That way, it keeps track of these cells as you measure it. That one, I'm not, oh, sorry. That was not done very well. But that way, it keeps track of it, and you don't make a double count on the same cell. Oh, I am not being very good today, am I? My hand. So sometimes you cannot quite see the boundary, and that's okay. Again, adding that noise helps you figure out where you've already counted a boundary, so you don't redo it again, right? So this one is just a partial cell, but it's clearly elongating. So I can still kind of look at it. If it was one that I wasn't sure, I would just let it go and not do it. Again, add noise as I go, right? So that's how you would keep track of your numbers. So I'm just going to do a couple more, and then I will show you what you can do to make an actual very quantitative measurement and count about these cells. So again, it's a good habit to process them and add noise, so you know for a fact that you're not double counting cells. Let's do a couple more over here. Right. Let's look at one of these that we weren't sure about, for example. Okay, so let's stop here. And so let's say, for the sake of the argument, that we have looked at all the cells at this point, and we have the numbers from every single one of the cells in here. We have 13. Let's do a couple more. So I'm just gonna do maybe So now you have all your numbers. You can now save them, and you can say save as, and I'm gonna say that these are my results for C3B. These are morphology results, right? These are not measurements. These are not anything other than your morphology results. I can now open it up. And now I can look at this, and I can specifically look for the roundness or the circular factor and use that as my guide. So I'm going to make that appear within the field of your screen. So in here, in this top bar, you would find these formats, and you would find choices to so in this top bar under home, you will find your sorts. So what you can do is take your data set, however many measurements you have, whether they are 1,000 or 10, it doesn't matter, and you can say custom sort and sort by the either the circularity or the roundness factor, and then add a level, and again by the round, smallest to largest, and click OK. Now, given this, you will see that you have a whole range of these numbers that are appearing, right? So here are these numbers. Now, we know for a fact that these 0.2 and 0.3, 0.4 are definitely going to be our cells that are differentiated or differentiating. And then the numbers that are higher than 0.5 are getting more towards the undifferentiated. The closer the number is to 1, the more boxy it was, the more round it was, and definitely those cells are going to be undifferentiated. So typically, we would take any number with has circularity factor of, let's say, 0.7 and above to be fully undifferentiated, and you can color code them in different ways, and numbers that are below 0.4 and below to be differentiating. For the rest of them, it would depend then, so they have the circularity and then the roundness. You can now look at the roundness factor and see if that helps you figure out some of those as well. So any of these where the roundness is still below 0.5, we can go ahead and we can place them into our elongated or beginning to elongate form. So they are not quite elongated all the way, but they are beginning to show some signs, and they are moving in that direction. And the numbers that are above that, 0.5 and above, would end up then towards your completely undifferentiated side. So this is a little bit more quantitative, a little bit more concrete setting where you can decide cells that are going to be categorized into your different categories when the morphological differences are such that you can easily separate them based on just their shape alone. So I hope this helped you figure out some more ways that you can use ImageJ to get quantitative, meaningful data out of your images when looking at cell culture model systems.